thank you for the introduction. My name is Anna, and it says on the slide that I'm here to ask you if you like food, but I think I probably know the answer to that. What I'm actually here to talk to you about is photosynthesis, which is taking carbon dioxide from the atmosphere, combining it with energy from the sun to create organic molecules. And this is the foundation for the majority of life on Earth. But despite the importance of photosynthesis, we have major holes in our understanding of this process. And so we need to fill in these gaps in knowledge in order to in order to create crop security and also possibly harness the energy from the sun in clean, renewable, sustainable energy processes. So in order to fill these gaps in, we need to have a more holistic understanding of the chloroplast, which is the compartment in which photosynthesis is housed in plant cells. This is a plant cell, and this green compartment right here is the organelle called the chloroplast. So we need to create an understanding of what's going on in the chloroplast in order to better understand photosynthesis. We estimate that there are about 5,000 gene products that function within the chloroplast, but the majority of the individual functions of those gene products are unknown. So take a minute to think about that. Thousands of puzzle pieces without defined edges, and without a clear picture or a clear place in the big picture. That really stymies our overall understanding of the physiology of what's happening in this compartment. So my dissertation is about building a tool to identify the edges of these puzzle pieces or the functions of these gene products within the chloroplast in a high throughput fashion. Traditional plant genetics rely on what are called gene knockouts, which is disruption of a single gene and then the observation of a phenotype or the difference that you can see in performance or appearance in the altered plant as compared to the unaltered plant. The problem with that is that genes from the same family often overlap in function, meaning if you just eliminate one gene, you're not always going to get a phenotype. So what we've created is an artificial microRNA library. And the power of artificial microRNA is they can target multiple genes from the same family simultaneously, thereby creating that hard sought-after phenotype. We're going to use our artificial microRNA library to identify calcium transporters within the chloroplast, which is a really important, calcium is a really important second messenger in plants. But this is not the only thing that can be studied with our artificial microRNA library. Any physiological process that happens in the chloroplast can be studied with this library. So we think this is going to be a really powerful tool and revolutionize plant genetics and enable us to secure the delicious, yummy food that we all eat every day.